we're going to be doing is we're going to be learning amino acids. These first three chapters, um, this would be the most important thing that we're going to be going over. Um, I'll send out a document so you guys can see what amino acids look like. But in this course, they at least want you to know the three-letter term. So they'll have three letters that signify what amino acid, what amino group it is. There'll be a letter, and then you want to know what's called the R group. So let's see if I can draw here in this thing. So the R group, and it's going to be really horrible drawing, but okay. C-O-O. Let's have it in this form. Okay. Have a hydrogen attached. We'll have this will be your amino group. Okay. So first off, this right here, if I can undo, this would be wonderful. Oh, okay. So this right here, we can see perfectly. This will be determining whether your group is protonated, whether your group is deprotonated, and we'll get into all that. But the reason we're starting here is that you have an assignment. It's the Henderson Hasselbalch equation, and this is where a lot of students can get tripped up, myself included. And even from time to time after you learn it, it's something that you want to keep fresh in your head because this is very pertinent to not only to biology, not only to biochemistry, but to different processes in our body. So let's say we want to know the pH in our the bicarbonate levels, or we want to know the pH in our blood, or we want to know, let's say something changes. The pH in our blood is 7.4. So let's say that bicarb changes that pH, or let's say we have a protonated version, or we have a deprotonated version. pKa levels change, so they change the acidity or the basicity of the environment. This could change everything, just by 0.1. So if we have a group right here that is COOH, this is the chicken right a little faster, but I'll just have it. This is the acidic version, okay? And I want you to take a moment and think, why do you think this is the acidic version? Okay. So in the acidic version of an amino acid, that means this is willing to donate. Okay. Let's be willing to donate. Now, if this was a COO group, and I might just search on Google if I'm not typing this a little or drawing this as fast, just so I can hop to it and get to the information for this class. Um, this would be the basic version. And think for a moment, why do you think this is the basic version? We'll just put BAS. Don't need to write. Why do we think this is the basic version? If we don't know, that's okay. Correct. And if you guessed it, that's fine. This version, deprotonated, D, DEP, where that's all we're going to put. Okay. So this Henderson Hasselbalch equation, pH is equal to pKa. And I figured that we'll probably have a more in-depth Q&A for this Henderson Hasselbalch equation. We can go a little bit more into math next time, as not for an introductory, Zoom meeting, but I want to start off with this one because this is what usually trips, like I said, this is what usually trips up a lot of students, especially when they see logarithms. If you're like me, I didn't even know what a logarithm was going into a neuroscience class, and I took it way too soon. And we had this logarithm for showing for the in and out of the membrane. And I remember going log log 1 og. What is this thing? I had no idea what that was. And once I finally figured it out, I thought two things. I thought maybe I might be in this class too soon, or maybe I just need to figure out what this logarithm thing is. And I did a little bit of both. Okay. So pH equals pKa plus log. Okay, we're gonna have base on top. Okay. Oops. How can I? Oh, cool. And I'll get better at this as we go on with this class. It's going to take a while to get used to drawing on a computer. Okay. So why do we think that this is the equation? What does it solve? What are we looking for? What's the reason for having Henderson Hasselbalch? Does anybody know? All our chemists, all our super smart chemists out there, I know you know the answer. Okay. So it lets us know the relationship between here and here based on the pH of the environment and the pKa. We're going to go over every single one of these. Okay. Okay, A minus, remember we go back over here. This is basic, deprotonated. If you can even read my writing, okay, right there. Okay. And then HA would be right there. Okay. Acidic. So when this has a proton, protonated, acidic is going to be on the bottom. Okay. A minus, which is not to get you tripped up for thinking that, hey, that's an acid. No, this is the basic version. Okay. Because it's deprotonated. It's missing this proton, deprotonated. What this would mean is the higher that the pH goes, the more it's going to be losing a proton. Okay. Okay. That makes sense. Yes. Okay. And then this version right here, acidic, the lower the pH goes, the more it's going to be willing to donate a proton. Okay. So, in short, solution with pH. This is usually is always this in the body, physiological pH. So, once you hear someone say physiological pH, we talk about 7.4. Okay. Okay. So if the pKa drops, this means what? It's very simple concept. So it just means it's more acidic. So H A up. Okay. And then you have to use your imagination. So I'm not going to draw that one more time. Basicity. Okay. It's going to be more basic if the pKa is higher. Does that make sense? Okay. And we're going to get more into this math a lot, not a lot later. We'll go into it next week. But I wanted to jump right into the hard portion of this class. This is where this is going to tell us everything for our biological system, for our metabolic processes. And I can go over every single one of those with you guys. We can go over step by step which enzyme does this, but we're not going to get to that right now. This is going to tell us, let's see if we can recap. This tells us, oh, there's an amino acid, which is this. By the way, this R group, can you see the mouse? Computer. Like that. Okay, so you can. Oh, cool. Has a little pointer. So this R group is going to define what amino acid it is. Okay. Yeah, I'm going to send you guys out a PDF you can print out, take it to the library if you have a printer at home, or if you want to fill it out as a PDF on your, iPads or something, you can use that. And I would suggest to draw. I would suggest to draw these things and make it a habit of at least drawing it once a day, going through all 20 amino acids, figuring out which amino acid is this, which amino is that, why does it do this? Okay. Because it may take a little bit of time if you guys are first seeing this. Biochemistry is supposed to be one of the hardest classes that you're going to take, even harder than organic chemistry, even organic harder than organic chemistry 2, Gen Chem 2, because it combines everything. It even has a little bit of physics inside it too. It has a little bit of psychology. Biochemistry is trickled in every single subject that you've taken for your undergraduate. And you may not be taking it as wanting to take biochemistry to go to biochemistry 2, or even to be a biochemist. But this equation here, acids and bases, is everything for OChem, everything for biology, everything for biochemistry. Learning this is so pertinent to understanding how our biological systems work. I can't stress that more than I already have. It's very important. This R group tells you the side chain, okay, of the amino. So it'll tell you, we'll get a little bit more into that later. Okay. If it's OH, acidic, COO minus, basic. Pretty simple. Okay. So we have this Henderson Hasselbalch equation. pH equals pKa plus logarithm. You don't know what logarithm is, I would suggest looking it up. Okay. Quick YouTube, find out how to do logarithms. Okay. We only have one assignment in this class that's going to be math based. And the equations, they're not too difficult. We can go over very similar ones like this next week. But for right now, the numbers that they're using, it's not too difficult. Okay. And then we got base on top over acid. This example question was at 7.4. Okay. Give you guys a second to digest that. And while we're on the topic, might as well give you guys an example. Okay. So I'll take this from my notes. There's when you get questions like this. Oh, hi Noah. Hi, good to see you. We're just going over some acids and bases before we go over the normal lecture. I figured this would be a good jump right into biochemistry. I think this is one of the most important things to understanding biological systems. Just understanding acids and bases. Okay. So pH equals pKa plus log of. Oh, you're good. You're good. It's gonna be recorded. If you're at work, you can hop in, hop out. Um, the first 10 minutes was just me shuffling, trying to get logged on through this. So you're chilling. You're good. Yeah, you're good. Okay, so with this, I want to give just a quick example, then jump right into chapters 1, 2, and 3. Okay, chapters 1, it's basically just going over kind of what biology is, what water is. Chapters 2 and then leading into 3 goes into aminos, which is kind of this right here, where we got the R group that's going to signify what amino acid it is, AA. Okay, so let us know what kind of environment they thrive in, what kind of environment they don't. They'll let us know if they're hydrophobic. Okay. But this pH equals pKa plus log over base over acid. We'll give you an example right now. Okay. So on the assignment, I'll make something similar to it. I don't want to give you guys all the answers or really any answers. I want you guys to work through it. I'll give you a very similar one. So mostly base and acids could be in it'd be in molarity, so moles. Okay. Sometimes you'll see molality, but it's mostly going to be moles. Okay. So let's say that the question says um, uh, conjugate base is 0. um, 0.5. Okay. Okay. We'll just call this B. And sorry for the horrible writing. Okay. And the acid will be the same at 0.5. Okay. I'll give you a second to just write out where this would go. Okay. So we have a pH, we don't know. We have a pKa, we don't know. But we have what the base is 0.5 moles. We have the acid at 0.5 moles. Okay. Give you a moment. And then remember, Henderson Hasselbalch, it shows the equation shows the relationship between pH and pKa. Okay. And while you're figuring that out, let me see if I can move this screen down. Oh, there we go. Okay. So we'll start it off. We're not given the pH, we're not given the pKa, but we're given the base. I shouldn't have put A, but A for acid and just this B for base. This took me 10 seconds faster. Okay. So we get pH equals pK. Oh, this is going to drive me nuts writing this stuff out. Plus, it's got to be a way to copy this. Maybe I can do this. I might have just figured something out. Oh, no, let's not do that. Sorry. Okay. Log. Okay, so then now we have the base on top, 0.5. Worst 0.5 in history that anyone's ever drawn out. Okay, and then we got 0.5 on the bottom. The acid. Okay. Remember, we don't need to be math majors. We don't need to be these knowing logarithms off the top of our head. We don't, but there are some that we do need to know. There's some that we need to just quickly know. Log 1. We need to know log 2, log 3, and what just quick math, we can know that something over something is more that of the same thing. So 0.5 over 0.5 is going to equal to 1. That's basic math. So really, this is just log 1. Okay. That's the worst writing in human history, but it's better. So 0.5 over 0.5 is going to equal to 1. Okay. 100 over 100 is going to equal to 1. 2 million over 2 million is going to equal to 1. Doesn't matter what that is. What we're learning is if the base and the acid on the top are the same, what does that mean? Log of 1 is going to be 0. Okay. And if you do know, if you don't know, that's fine. But I would definitely recommend to look into a chart. I have something, if you really want me to, just email me or I can put it in the chat. But really, you should know a good idea of what log 1 through 10 are for most math classes, past calculus to past. If you don't, it's fine, but it's going to equal somewhere between 0 and 1. So forget log 1, that's basically the starting at 0. I try to remember at least 1, 2, and 3. So log 1 would be 0, log 2 would be 0.3, and log 3 would be 0.5. And you can just remember by your fingers. So I just remember log 1, 0, log 2, 0.3, log 3, 0.5. Everything after that is going to slowly inch towards equaling 1. Okay, because log 10 is going to equal 1. And hopefully that wasn't too confusing. Just try to make it as simple as possible. But I remember log 1 is going to be equal 0. Log 2 is going to equal 3 because the next finger is 3. Okay. And log 3 is going to equal 5. Okay. Sorry, 0.5. 0.3, 0.5. Does that make sense? Okay. Got it. And then this would just let us know that this relationship between the pKa and pH is the same. Okay. That's all that would let us know. This is 0. These cancel out. pH equals pKa. Done. That means we'd be looking at a Zwitter ion. It's Could be equally as protonated as deprotonated. And if you made it this far, that's a very similar equation to question one on the Henderson Hasselbalch homework assignment that you guys have. Okay. But it's not too difficult. When I went over it, I just wanted to refresh it and keep it fresh in my head. Towards the end, if you don't know the logarithms, you might want to have a calculator. That's totally fine, unless you're planning to go into medicine. Then yeah, know the logarithms. But if you're not, that's fine. Nothing against that. Learn them, but there are definitely other things like I would rather toss in your brain than log six. Like it doesn't make sense to me. Okay. Now let me share my screen so I can go over quickly. I don't know how much time before this cuts out. Trying if I could share my screen to show the PDF. But if you have any questions on anything, chapters 1, 2, and 3, toss it in. You can unmute yourself, put it in the chat, email me. I'm here to help always. Dr. Baldelis, she's here to help as well. We have so much help for this class, and just buckle in. This would be if you put in the time, we'll notice it, and this could be a very difficult class. I'm not gonna beat around the bush. Um, how do I share this? Sorry, I'm the worst at this stuff. Sharing your screen will close the whiteboard for everyone. Okay, I'm gonna close the whiteboard. Everyone good? Good. Thumbs up. Okay, good. And let's see. Let's go to this. Try to remember how to full screen. Command this full. Oh, there we go. Okay. So I'm gonna jet through this as much as possible. Because I noticed some of you guys took the first quiz, which is fine. There's I believe three quizzes this week. One of the first ones is on plants. That's not my forte. So I'm completely gonna be skipping over. I see some chuckles. That's not my forte, plants. So I would advise asking anyone but me about anything about plants. But I'm going to go into just quickly go into membranes as long as the Zoom is here. As long as you want to stay here, that's fine. I could talk about biology, biochemistry for 24 hours straight. Okay. Okay. So I took a lot of screenshots from this, and then halfway through, I add kind of my personality to teaching. Just to be able to get the information through. I think that learning through mnemonics, learning through making something funny and silly, it makes it stick in your head. You can think about it, 2 months later. Um, for example, it took me a while to off on a tangent. It took me a while to remember independent and dependent variables. I remember going like mix them up. Oh, what's this? What's this? And if you're thinking about it, if you're listening to it, you might be like me and going, hey, I mixed this up, or you might just be a normal intelligent person unlike myself and already remember them. But I remember this silly thing that one of my instructors told me, and she said, if I just showed up with purple hair, would that make everyone's heartbeat? Would it make their heartbeat go up or go down? Would that change everything? And we went, no. She goes, okay, well, that would be this variable, and then if your heart rate will be this variable. And so, not going to go into that tangent, but that would just be dependent, independent variables. But silly stuff like that, I remember thinking like, yeah, just someone changing their their hair color is not going to make my heart rate go up or down. So it's not going to differentiate. Um, that's not going to give me any results. So just silly stuff like that. Off on a tangent. Okay, so membranes. Um, the cytoskeleton, I just highlighted a little portions of this that were important. Active filaments, intermediate filaments, and microtubules. And you can put in the chat, you can talk with me, you can just hang out and listen. But do you guys know what this does? Like just the basicity, the basic portion of what's the point of having microtubules, active filaments, intermediate filaments? Yeah. They help for transport. Structure, yeah. Some mobility, some movement. Structure would That's right on. It keeps everything packed nice and tight. As long as we understand that, we understand this little sandwich of two membranes. We understand this big thing for mitochondria, and you got big arms in there, Noah. So you got probably 47 different mitochondria in one piece of your form. So, so yeah, like microtubules, they're they help for I can put this simply. They help keep everything the shape, and they help with movement. Okay. So the cytoskeleton, as I highlighted here in this little portion, think of it as a highway. So it's like getting on, I don't know what the local freeway is for you guys. I'm here in California. One of the biggest ones, if you ever Google like a 405 freeway, it's very polluted. So this would help shuttle these molecules up, down, left, right. They're trying to go across the state. They're trying to get on a different freeway. That's what that does. Okay. It's just these little portions. And I'm going to send this PDF to you. It's really just highlighted for the most, for the biggest details. Okay. So we're still in chapter one. Anaplastic reticulum. There's two portions. There's a rough ER and a smooth ER. One of them processes drugs. One of them helps with transport. It's really a smooth. Rough, it's going to be studied with ribosomes. Ribosomes help with, we'll get into it later, but it helps um, for your DNA to RNA to protein. It helps for transcription translation. That's where that site is. We'll get into that in a moment. Okay. But mainly this whole point, I just want to just show you just a little picture. This rough ER, these little studded things. Can you see my mouse? Again, just checking. Okay, rough ER, these would be these little ribosomes, these little green dots. Boop. Lumen. Um, I would say this is a good point to stop for a second. Lumen, it's just the inside of the ER. Okay. So it would be, think of the ER as just anything. Okay. And then the lumen would be what gets inside. And that's gonna be very important because that portion, when ribosomes are attached to that ER, the processing of drugs. So let's say you drank some alcohol, it's gonna get processed by going inside the lumen. Okay, and then it's gonna go to the liver, and we're about to get into all that. But that lumen is that space that's inside. Okay. Oh, I already had it in the notes right here. Okay. Okay. So forgot about the portion of the notes. So the lipids for here, that just little checkpoint, I'll read it with you. So lipids, they form phospholipids and cholesterol. Phospholipids, they're for cell membranes, so they create the cell membrane, the outside of the portion, right? And then cholesterol creates steroids, testosterone, estrogen, androgens. Big thing here, okay? If we notice this mouse working, always gotta check. Okay, big thing here. These 1, 2, 3, 1, 2, 3, 1, 2, 3, boom, and then we got this fourth one right here. Okay, if you could just stop for a second and think, what would be the point of having cholesterol to create a steroid? Okay, cholesterol, I'll let you think for a second. What will be the point of having cholesterol creating these steroids? Estradiol would be estrogen. Testosterone would be obviously what's filling our body. Okay, well, cholesterol, it's fats. They're hydrophobic. So if cholesterol is made from fats, and then they branch to make testosterone, estrogen, estradiol, steroids, wouldn't that make them hydrophilic as well? It would. Yeah, it would. So there are these hydrocarbon groups that make them, like I put right here, they make them hydrophobic. So they're not wanting to. Oh, here's a better picture. They're not wanting to sit on the outside. So this outside, yeah, there we go. This outside layer is where all the water is. They want to be shoved right in here, and they'll reposition. So I can get this little portion right here. They'll reposition this cholesterol hydrocarbon tail in the middle. Okay, because the outside of this is all, let's just say liquid, right? Water on the outside of these. These little circles right here, circles, water, water. The inside, this is where all this hydrophobic area, amino acids, steroids, they want to be pushed right in the middle of this area. Okay. So cholesterol will be right here. But the weird thing is that they'll have a polar hydroxyl group. So that means that they'll stick to this upper layer, and then they'll still have this layer in the middle. Okay, just want to touch on that a little bit. We're going to go over this portion like 100 times in this class. So hydrophilic, they love water. Hydrophobic, they're afraid of it. Get me the heck away from them. Okay, so cholesterol wants to be right here. You want to be best friends. Okay, you want to be away from water. Get me the heck away from there. Okay, temperature can change cholesterol. I'm sorry, temperature can change the rigidity. So if we have temperatures way up to the sky, that makes it super rigid, makes it super packed. Okay. And if the temperature is very low, then it increases the fluidity, makes everything kind of just flow a little bit better. Okay. We got some more stuff on the ER. Lumen, so like I was saying earlier, ER during translation. That's really it. Boom. Lumen is this little space between here. Okay. So another thing, I'll go off on a tangent. I think it's going to cut off in 2 minutes, but protein folding, we need chaperone proteins to help fold it into its 3D shape. That's how it gets this protein. There's a step called proteolysis. There's phosphorylation, which we'll learn from amino acids, glycosylation, ubiquination, liquid group, or sorry, lipidation, prenylation. What's the point of this? Okay, all of this happens post-translational. It's a post-translational modification, meaning that it adds a carbohydrate, for example, so it can have proper folding. So as we look at this next picture that I made here for you guys, okay, this whole little diagram kind of explains once you get a protein, what the heck is going to happen. So if there's phosphorylation, we'll see amino acids like serine, we'll see threonine, we'll see tyrosine. That could be phosphorylated. Why? Because they have a hydroxyl group. They have an alcohol group that's on the side chain. Remember, if we're going that R group side chain, S, T, and Y, it has an OH group. Okay, so that means that it can hydrogen bond. Okay, and then I made a little chart here for you. So STY, these are OH groups. Okay, glycosylation, all that really means is just glyco. Just break it down real quick. Carbohydrate. Made little notes right here. Gives the protein a cute little name. So the protein gives it a little carbohydrate. So it goes, hey, don't touch this thing. Okay. Ubiquination does the same exact thing, but it marks it so it can be destroyed, be broken down, that cost. And then this one right here, lipidation, just attaches the lipid group. So proteins, after they're created, they will go through a process of either phosphorylation, where they, can bond, hydrogen bond. They'll be attached with a carbohydrate, glycosylation. They'll be tagged to, hey, get us out of here, right? Or they'll be attached with the lipid group.
Okay, so why do we think that this happens? Okay, so plus and minus, they'll attract. So unlike charged. Okay, so this Na plus and the Cl minus salt, when it's placed in water, oxygen will surround the Na plus, right? Like this. Why? In OChem, you'll learn that it's slightly negative, and H is slightly positive. The theme would be is that opposites attract. So if we look at for here, oh, you can't see my mouse, so I'm doing that. You only see on this side. That's wild. Okay, so if you look at it through here, oxygen on here, the more electronegative they are, right? Hydrogen is going to be on the least side of the electronegativity. So if it's low, it's going to weakly attract if it's low electronegativity like these hydrogens, it's going to have a weak attraction for electrons to bring them in. Okay. Just this simple little picture, just to try and explain this. I know it's not too much in depth, but it's more so just to put a visual to everything. Okay. Brownian motion, we'll see these little dots right here. When we really see this clear water right here, we'll see all these dots. Really, the water for Brownian motion. Um, it's just water and glass looks like it's still, but they're constantly colliding at extreme speeds. So this is really what you're looking at when you're looking at water. Okay. Okay. So just to recap, oxygen is strongly electronegative. So that means it wants to attract the electrons to itself. So if oxygen wants to attract, these O groups want to attract the electrons, it wants to hold this right here. Remember? Now, if this is weakly electronegative, it doesn't want them. Okay, so it's just going to pull this oxygen group. Okay. Makes sense? Up to the next section. Okay. Um, this is kind of gonna go a little bit more into depth as well, but hydrogen bonds, boom. These are just two different different things we'll see in DNA. We'll see this in protein. We'll go over this 100 different times. But this is just to put the visual in your head. Adenine pairs with thymine. There should be two hydrogen bonds. H bond, see? Guanine pairs with cytosine. Three bonds, three hydrogen bonds. Boom, boom, boom. Okay. This little memory concept. Um, I had a teacher teach me this one time, Funkelbrisk. I don't know why. It sounds so silly, but it does stick. It maybe you got to say like four, five, six different times. The other portions doesn't really matter at least right now. It goes by electronegativity. But FON, if you just remember, those are the only elements that are a hydrogen bond. Okay. F right here, O, and N. That's it. Okay, fun. FON. Coal or chlorine, Br, bromine, iodine, so on and so forth. Just follow it down. Okay. So what was happening in this water right here? This picture, we see the water still, but it's constantly moving through Brownian motion. Okay. When something is placed, so let's say something that is nonpolar, it's first placed, it's going to have these water molecules surrounded right here. This is going to be a non-spontaneous reaction. Okay, this isn't naturally that you can read this like the descriptions of this later. I know there's a lot of math, a lot of random stuff on here. I put it on here just if I had time to go through it. I'll kind of burn through what's important. But these little waters right here that's around this nonpolar molecules, let's imagine this is oil. So if you've ever had you probably have a frying pan, you put, well, let's say you left water in the pan for some reason, you put oil. The second you drop oil in there, the water is going to disperse and go, hey, we don't want to be around here. But let's say if you disperse it where one drop of oil, water would be surrounded by both of them. Why? Just think about that for a second. It would surround them because the entropy would decrease, and the entropy would decrease because water would surround this thing and go, "Hey, we don't like this oil that's in here," and the other oil droplet on the pan is going to go, "We don't like this either," and they're going to slowly merge into togetherness, and then they're going to go back to spontaneity. It's going to be back to spontaneous. Spontaneous just meaning that it's naturally supposed to occur that way. Non-spontaneous means that it needs to have some kind of outside influence to work. Okay, so there's this whole chart right here. I wouldn't think too much into this other than this is just a way to show you how it works, right? So if it goes up right here, boom, then the products are created. It needs some kind of activation energy for it to reach this product. Okay, you'll probably see this maybe one or two more times in this class, but this is just to introduce the idea of it. Okay, concepts. Um, we can go over this. I guess I'll leave it to you if you want me to. I could break this down super simple, or we can skip it. Thumbs up, thumbs down. If you want me to go over the like a concept check for this, thumbs up. Okay, I'll make it quick. So if there's two hydrogens and it swaps over to 2H, the delta H for this is going to be non-spontaneous. Without having any background, or maybe you do, maybe you don't on this, why do you think it would be non-spontaneous? Non-spontaneous would mean that it needs to have an outside influence. Spontaneous would mean that the reaction needs to be um, spontaneous would mean that our biological system naturally leads to being like that all the time. Okay. So in here, we see that the energy must be added to break chemical bonds. Okay. Endothermic process being inside. Okay. So if there's an energy that needs to be added to break these bonds to make them 2H, the visual would be they're stuck together. Now there's HH. Okay. So if it needed to have energy in there to break them down, that's not a natural occurring process. Eventually, but it's not. Now, if we have these where they form a chemical bond, right? So this would be 2,2. And also, side note, when we see 2H, think of it as like, yes, we'll add them together in a math equation, but it'll be just like having, oh, how many things two? Oh, be like having this, two water bottles. Yeah, that's really all it would be. So, when they form a chemical bond, imagine they just merge and make this bigger water bottle somehow. Can't do it without having energy, right? Um, but they would form this bond, and through the forming of that bond, it will release energy. So the reason I put this here, I know this may be like, what the heck is he going on? I just wanted to put a visualization for when there's a bond that's created and there's a bond that's broken. Our body either naturally will do one or will naturally do the other. So when a bond is created, it has to release something. Okay. When a bond is broken, it has to have energy. Think of it as something goes and grabs it and rips it from there. Something has to come out and rip it. Okay. If a bond is put together, it makes it spontaneous because energy is released. When there's a release of energy, it makes it a negative number. So I mean, something's leaving makes it a negative number, spontaneous. So I know that was a mouthful, but I would just remember that when a bond is formed, energy is going to be released because that is one of the biggest things to understanding biological systems. Okay. Um, that's how ATP works in our body. Like you go to the gym, you're working out, you're struggling, you're building up lactic acid, right? Lactate, really. They call it lactic acid, it's really lactate. But when ATP is formed and used, it's a break between two phosphate groups, breaking of the bond. Okay. When it's formed, that means energy is released, ATP is used. Okay. That and for I hope that makes sense. And if it doesn't, that's fine. It may not be explaining it that well, but it's the form of those two bonds that releases energy, and that wires our entire biological systems. I'll maybe think of another way to go over that in a moment. But I want to get to a couple different things. Um, chapter three, these were you could tell by my tone with this. I wouldn't say that you need to know the typical pKa's of this. That I don't know them. I really don't. I mean, other than histidine, lysine, arginine, yes, of course, which not important at this moment. But these are your protonated amino acids. Okay. And I put a little picture right here for lysine. This is what lysine is. This is what arginine is. This is what histidine is. Guanidinium group. We'll see that, and so on and so forth. Okay. This purple, this purple box right here. We're gonna see that right here. We're gonna see it right here in histidine. Just a visual. I wouldn't freak out. I wouldn't go, oh crap, I need to know this stuff right now. This is everything we're talking about, but they're just having another way of showing you, another way to confuse you. So we started this class in the very, very, very beginning where I said, oh, COO minus group means, if you remember, means it's deprotonated. Deprotonated in the green. Okay. That means it's super basic. It's higher than 7. 7, 8, 9, 10, 11, 12. This is super basic. So it means it's positive, right? This is Witter ion, meaning that it's not really basic. It's not really acidic. Okay. But if we go back to this side, I'll explain what this is if you really want me to. But this means that's protonated. Okay. That means it's under 7.4 physiological pH. That means they need to add on an H group. Okay. So now going back to this equation, at pH of 0, everything would be dead. But let's just say pH in this example. It has an H group, right? Because it has to make up for that pH environment for the concentration. Okay. As we increase the pH, we notice that this H group goes away. Whoa. Okay. Now as we go more, we notice. What do we notice? What was the change from here to here? Lost another H group right here on the side. So the higher the pH, it's more basic. The lower the pH means more acidic. This is just a visual of it. So I know at first it may be like, okay, well, what are we looking at? I wouldn't worry about these right just yet. You're going to learn your amino acids, but understanding this concept, I keep throwing it and throwing it and throwing it and throwing it at you guys because this tells everything. Like at a certain point, you'll be able to say, okay, well, we're looking at this amino chain. It's got these letters. Oh, well, it has this letter. Okay, here it has it in this direction. Well, it must be hydrophobic or it must be in an acidic environment because it has that on its side chain, right? That's why I keep going back to this. Okay, COO minus, COOH. Okay, hopefully that made sense. If it doesn't, oh, I'm sorry. Um, this is from Jack Weston. A lot of the stuff the pictures that I get are from Jack Weston. Um, they're like a prep company for I think they help with some nursing programs too, but it's mainly to get people into med school. But their tools that they have is great. A lot of visuals they have is great, and most of their program is free. I think it's Jack Weston is phenomenal. Khan Academy is phenomenal for learning biochemistry, chem. Good news and bad news. You will need to know these. I don't want to say like the back of your hand, but you will need to know these amino acids. But if you look at them every day and you draw them and you just tell yourself, okay, if you don't know them, at least if you don't know them yet, you're gonna learn them. It's not that difficult. I'm not this super intelligent person. I'm not and I know them. And there's some people that know them by the back of their hand. And hey, I commend you on that. Sometimes I forget I'm human. But I would say at least being able to put them in their groups of I'm sorry, of nonpolar, of polar, of whatever kind of side chain there are, positive or negative, then you can understand what kind of environment they are. And this all the chapter one, chapter two, the beginning of this, learning Henderson Hasselbalch led up to this. And I get it. You're like, oh, this is so much. Like, why all this? Well, the first two chapters really are supposed to be review, but they're bringing into this. And this is the most important thing. Every slide I've showed you, this is the most important thing. They all led up to this. Okay. Proline has a side chain that helps folding a protein. Not going to go into it anymore. Okay. Glycine, it has this. Let's see if I can zoom in a little bit. Glycine has a side chain that's the only one that's achiral. Okay. Same H and H. All these blue groups, by the way, these are all side chains. Okay. You don't know them. You don't know what their names are. And that's fine. You just look at them, or you go to the bathroom, or you're walking to other classes while you're at work. They will come. I promise you. It's knowing your amino acids show by how many times you put them in front of yourself, not by sitting there and trying to memorize them in 10 hours in one spurt. Like, that's ridiculous. I mean, it might work for some people, but not for most. Alanine, CH3, valine, think of it as a V. Boom. Okay. Leucine, very similar to isoleucine. Um, and since you work out, I'm going to keep picking on you because you get massive forearms. Isoleucine, leucine, and valine, these are branched chain amino acids. And I remember I got a question on well, I shouldn't be saying I got that exact question, but I'll paraphrase. I got a question on one of that for just learning cat stuff, if you get what I'm saying. We're saying, oh, what are BCAAs? And this branched chain amino acids. Why would you call them branched chain amino acids? Because they branch, because it's a branched chain. Okay, this chain of carbons, it's really a carbon chain. Carbon, 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 carbon. Okay. And we see that with this carbon, carbon, carbon. Oh, sulfur. No, that's not it. That's not a branched chain. These chains branch this way. Isoleucine means isolating this part right here. Leucine is the same thing. It just has a branch right here. Okay, so 1, 2, 3, 4. 1, 2, 3, 4. Isoleucine, it's kind of by itself. That's really it. I wouldn't put too much emphasis into memorizing exactly what I just said. Just become familiarized, draw it out. We already went over these. STY, STY. And I got a great mnemonic for all these before I go into the polar. Non-polar, it's GAV-LIMP. So G, glycine, A, V, LIMP. And then you can add in FW as well, which is phenylalanine and tryptophan. These are all non-polar. I don't want to go any more than that to add in a ton of information, but GAV-LIMP FW. I think I might have it in the notes somewhere down here. If not, I would I would definitely write that down or grab it from the link. Polar, we got serine, threonine, tyrosine. These can all hydrogen bond. See, it's right here. Boom. You got a hydroxyl group. What is a hydroxyl group? Hydroxyl alcohol, hydroxyl, hydro, oxo group, OH group. That's all we're talking about. They're just chemists trying to confuse everybody and have their own language. Really all it is. OH, OH, OH. These are the only groups that have that. Oh wait, what about tyrosine? Isn't this one? And it's the same thing. Okay. So don't want to confuse you. Just know it that sometimes people place it in nonpolar, some people place it in polar. That that's a nuance for later. I wouldn't worry about it right now. I would worry about where are these sections at? Okay, it's over there. Okay, cool. Great. Just notice it, move on. STY. Okay. Cytosine. Oh gosh, super important. So important. This creates tertiary and quaternary structures. If you break good luck breaking a cytosine bond. Like, I'm sorry, a disulfide bond. Good luck. Very difficult. But if you can break a disulfide bond, you're breaking this SH. Let's see if I can, maybe I can type right here. Can I type? Okay. Right. Or maybe I can go back to the board to draw. Yeah, whatever. Okay. Doesn't really matter. I would just remember that cytosine can bond to itself. And then it loses the H group, loses the H group because it bonds together. So it's an S bonded to an S. That creates on the side you see here these disulfide bridges, and so important. I would just leave it at that. You'll hear that like disulfide bonds so many times. Asparagine, glutamine might mix up with aspartate, glutamate. Aspartate is aspartic acid, glutamate, glutamic acid as well. You may see it named as that. These ones you may mix up, and that's okay. I would just remember D and E. D and E is negative. Eventually get there. Um, Positively charged. I used to have a picture online that I made to just remember these, but it's at this point just bury it in your brain. Positively charged. That just means, remember going back to Henderson Hasselbalch, it's got well, two things. Not only it has nitrogens, nitrogen, 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 nitrogen. But that means once it's at physiological pH 7.4, it's going to still be plus. It's going to be a plus group. So maybe that might not have stuck. But at 7.4, these are still going to be protonated. Okay, it's still going to have a plus group. Okay, if you remember, I highlighted histidine. Well, histidine is the one that's not going to have it because it's at 6.0. So, but they're positively charged. I would just remember that. Lysine, arginine. Okay. Uh, go. That's why this one is circled. So we can even read the description. So unlike other amino acids in the group, it's left uncharged. So these two, they have a charge. But if the pH in the group, they're like, oh, hey, like pH is 7 or 7.4. Is the histidine in here? Is it going to be positively charged now? That's just because of the environment. Normal environments, let's say if it's under there, yeah, it's positively charged. These other amino acids are not. These are the only ones that have a positive charge. D, E, aspartate, glutamate, negatively charged. So if they're in the same situation, negative. And then nonpolar, these are just the aromatics, aromaticity. Aromatic just means that it just think of this ring. That's what I would think about it. it's really, I would just leave it at that. Tryptophan, this looks like a W in other pictures. Tyrosine, it's like a Y. Um, I know it's quite a bit to look at, but those are, I'll give you a little cheat sheet to just work on those. That'd probably be the best use of your time for this class to learn them. Everything I was just saying, R chains, carboxyl groups. We need to go over peptide bond formations. This is kind of going crazy. But I wanted to get over here. This is kind of like my personality hopping into the class now. So I want you to stop and think, kind of random, but protein moves from, I'm sorry, to create protein, it goes from DNA to RNA to protein through a form of transcription translation. Um, Thumbs up, thumbs down if you understand that portion. Well, I'm getting my charger here. Okay, somewhat, yeah, good. So how would we be able to create more DNA if we use DNA to create RNA to create protein? Okay, so eventually, like, uh, wouldn't we run out of DNA at some point? How would we create more DNA? Gotta move this to charge my phone. Or my laptop. 1 second. Okay. And then we got the young Sheldons, they're sitting there together. But I brought this in just to kind of tie everything in together. Okay. So here in transcription, we go from DNA, process of transcription to RNA, process of translation to protein. Okay. There are some ways you can go backwards, but we're not going to go into that at this moment. But if we're constantly converting DNA to RNA, are we ever going to run out? So what if every cell is used up its DNA to make RNA? Okay, I got the oops. Okay, so this is the picture of the process, okay, for transcription, but it goes through replication. Okay, it just does it again, it does it again, does it again. Okay, this happens inside the nucleus. Okay, that's where that's what it uses, and uses DNA polymerase to create more DNA. That's where this portion happens. Okay. But then when it creates more DNA, it goes, okay, transcription, same thing in the nucleus, RNA. Then it wants to convert RNA to protein, it pop it to the ribosomes. Okay. More so just a picture, just get things jogging. If you haven't seen it, they'll come in the chapter, but just to bring it up ahead. Um, I know I got a lot of these ridiculous things in here, but it's just these 3 chapters are your biggest ones. So I just want to make it a little bit lighthearted. On top of that, just to get the brain jogging. Okay. Okay. So here's Hulk Hogan's little segment. Okay. So Hulk Hogan asks, where did the M in mRNA come from? Oh, well, I didn't catch that. So let's see, RNA, and then it goes mRNA. Where did that come from? At least getting an idea of what this portion is will let us know how protein becomes protein. So everything we just looked at were amino acids. Amino acids, you tag them together at certain points, they eventually become protein in a way. So, well, where does this mRNA come from? Okay, it's a messenger RNA. That's really all it is. It has a cap, a 5' cap. They'll have a poly A tail, so think of like a ponytail, and they'll have a splicing. Okay, you can look at the pictures in the PDF on your own time. But there are these exons, these things right here, and introns. Okay, so put them in red, red, and then green. Green stays, red cuts out. Done. We don't need it. Splicing pulls these introns out. We don't need them. Boom. Now it creates an mRNA. So in this portion right here, go up to this picture. Transcription, RNA, translation. Let me go right here. Okay, remember I was saying it occurs in the nucleus. So there's an enzyme, RNA polymer. Enzyme does this. Okay, it catalyzes, meaning it speeds up the process of DNA to RNA. Catalyze meaning makes it so much faster, it's ridiculous, like tens of millions of times faster. If it didn't have it, we'd all be dead. It wouldn't happen. Okay, and then it becomes mRNA. It becomes mRNA because it's a mature RNA now ready to be created to a protein. Okay. There's some stuff in here. Let's go over this last portion. So it says, how can I put it here? So cancer, rhinitis pigmentosa. It's just kind of see fuzzy, like fuzzy-ish. Um, you almost can eventually go blind in your eyes. It's kind of similar to stigmatism, but it just keeps getting worse and worse and worse. Or muscular dystrophy can be caused from failure to cut out these introns. Why? Maybe maybe just try to think, why would these, so I can reword this question differently. These diseases in the body are caused from not being able to cut out these introns. Why? Well, the finished mRNA, so let's say if these stay, then the protein will have these introns, so they're non-coding, right? They cut these out because they don't code for any kind of DNA. Now the DNA has them in there. Now they can be translated into protein. That protein's done. It's nothing anymore. That's how you get some forms of, I mean, one way, right? You can get cancer, muscular dystrophy, so on and so forth. So these introns are so important to get rid of. That's why we see Hulk Hogan taking a shot. I know it's kind of silly, but just trying to. If we just look at it like that, like we can learn tons of information a day, and it's like the information we can learn a day is it's uncapped. Okay, so introns, don't think of it as they stay in the DNA; they're leaving. We don't need them anymore. Exons, we pull them together. Okay, so exons are joined, introns are cut out. Water. I know we kind of went over this earlier, and maybe if I would have had this picture, it would have jogged my memory. So there's a slight negative and a slight positive between H2O. So that's kind of important to remember in that picture with them. This is one of the things that Dr. Cordelis has for her PowerPoint that she went over for the class. Just kind of telling like, oh, like interactions, covalent, not covalent, hydrogen bonds, van der Waal interactions. I'll leave that in there. I have some notes in here. I'm not going to go over them, but the simplicity of it is that an iconic bond, I'm sorry, an ionic bond is Na plus and Cl minus, the charge groups. That was this portion that was way up here. As we trying to add information later, my OCD try to add a little bit more information into this to make it make sense. So, oh, why is this split up? That's why. Yeah, that's why it'll be evenly split. Mm, okay, so typically metal and a non-metal. If they gain electrons, they're anion. If they lose, they're cation. It's cool. We already went over fun, FON. That's a hydrogen bond. Um, van der Waals. Van der Waals interactions, they're so important. It's one of those weird random names, Van der Waal, that you kind of see once, you never forget because who do we know that's named Van der Waal? I mean, if you do, hey, hats off to you because I don't know anyone. um, they cause temporary dipoles, and if you don't know what that is, that's fine. If you do, then I'll jet through it quickly. But a temporary dipole is just like they're this bond, and let's say we only need it to be a bond for a certain amount of time. That's really it. Boom, done. It's not stuck there forever, permanent. It's not, hey, it's not like these disulfide bonds where it's like so much has to happen to rip these things off. These are like they are married. Van der Waals are like, we kind of just went on a Tinder date and nah, and oh, they're just go back to what I was saying a couple seconds ago. Okay, so van der Waals, it's kind of just temporary dipoles. They have it as temporary, in temporary dipole meaning that electrons move around. So they have it as temporary because DNA replication, it opens, it closes. It opens to replicate the DNA, it closes to protect the gene. Hormones, they'll attach to receptors, they'll take off from receptors. Okay. There's one more I put in there. Oh, enzymes, of course. Enzymes have to be reversible. Um, and they also, after an enzyme is used, you'll replenish itself. So it's very cool. Picture of covalent bonds, ionic bonds. Ionic, it just goes, hey, I need everything. You're selfish. Covalent bonds, they share. These are married. This is the Tinder. Okay. Okay. So then we got acid base. This is what is the name of this? You got the names of this. You can quiz yourself. If you don't know them, that's fine. Remember base is a negative. Acid has an extra hydrogen, has an extra proton. Acetate, acetic acid. I'll put a picture of them right here. And then where we go? Oh, eukaryotic cells. This is where transcription happens. So we see that it happens inside the nucleus, and then when translation happens, we see it's outside in the cytoplasm. Cytoplasm and in prokaryotic cells, you see it here. I got some questions here. You can quiz yourself on. We'll have the answers underneath it. I have it twice. Not sure why I have it twice, but you can go over the answers twice. Super important. Okay, there's a lot, but these first three chapters, I wanted to make it a point to go over the nuances. The other stuff should be pretty basic. If you have any questions, you can just send me an email. You can send Dr. Pagalis an email. You can send any one of our helpers here an email, but I'm here to help, and just want to make sure that you guys succeed in biochemistry. It's a fun class if you make it. It can be quite a bit of information, especially if we're going towards the metabolic pathways. Um, but just remember that you have support. You can always ask questions. But um, for me, I really look at effort. So if I notice that someone is not getting something and they're trying, and they're putting a lot of effort in, that doesn't go unnoticed. Okay. So understanding these biological processes are very important, and I take it very serious. Not only because this can help save someone's life if you guys decide to want to go into the medical route, or the veterinarian route, or any kind of pre-medical field, right? Even podiatry. Let's look at feet. I'm just kidding. Yeah, it it will help somebody someday understanding the processes of the body, and maybe not knowing actin filaments in this, but understanding actin filaments allows us to know that their structure. And then if we go, okay, well, why do we need to know the structure? Oh, well, because if there's missing this portion in this situation of their body, then we need to know that. But the biggest key takeaway is that we need to know pH. We need to understand amino acids, and a change in amino acids, a change in one amino acid can change everything depending on where it's at and depending on the environment as well. Okay, so good luck to everybody. If you guys have any questions, just email me. I'll be here next Thursday. I know I didn't put a time, and we took a little bit longer. I'll keep it a little bit more structured next Thursday for everyone. But I hope you all have a great weekend. We're getting near the holiday season, so put the time in now. So if in case you need more extra days off later in the year, in the semester, you guys have the time to do that and not worry about cramming work assignments or cramming knowledge. But this is fun stuff at this point. We've made it this far, so congratulate yourself. But the work starts here. Okay, have a good one.